Hey Pharmatalk subscribers, it's your girl, back with another dose of pharma knowledge. Today, I want to dive into a crucial topic, out of specification results. Now, what exactly are they? Simply put, an OOS result is a result that falls outside the predetermined specifications for a test. This could be during quality control testing of a finished product or even raw materials. When this happens, it's a red flag and triggers a thorough investigation to find out what went wrong.
I can explain the difference between OOS and OOT. OOS stands for out of specification and refers to a test result that falls outside the predefined specifications for a product or material. This means the product does not meet the required quality standards. On the other hand, OOT stands for out of trend and refers to a test result that, while still within the specifications, is unusual or deviates from the expected trend based on historical data. It's like a warning sign that something might be changing in the process and could potentially lead to an OOS result in the future. Essentially, OOT is a proactive measure to identify potential issues before they become out of specification, while OOS is a reactive response to a result that has already failed to meet the required standards.
That's a clear explanation of how to determine out of specification and out of trend results. Using control limits and specification limits along with statistical analysis helps identify any issues in the production process. Plotting the data in a range chart is also a useful way to visualize any deviations. Monitoring those 5 successive data points outside the expected range is a good way to detect out-of-trend situations. Out-of-specification events can arise from various points in the production and quality control processes. One common source is laboratory error. This can include mistakes made by analysts, incorrect calculations, malfunctioning equipment, or problems with sample preparation or standards. If a lab error is confirmed, it doesn't necessarily mean the product failed, but it does indicate a need for corrective action to prevent future errors, such as better training or equipment maintenance. Another potential source is a lack of precision in the test method itself. Even validated methods can have inherent variability, leading to results that fall outside the specified range purely by chance. If this is the cause, it doesn't mean the product failed, but it does raise questions about the reliability of the test method. Another category is non-process-related errors in manufacturing, which include human or mechanical errors during production. Examples include forgetting to add a component, equipment malfunctions, or cross-contamination. If these errors are confirmed, they usually mean the specific batch of product has failed. Finally, process or manufacturing problems related to process control can also lead to OOS events. This includes issues like incorrect mixing times or uneven blends. Confirming these problems means the affected batch is a failure, and it may also indicate potential failures in other batches as well. Category 1, laboratory error, involves various mistakes that can occur during testing. Here are 3 examples. First, an analyst might misread a measurement or incorrectly record data, leading to a false OOS result. Second, a piece of lab equipment, like a balance or a spectrophotometer, might be malfunctioning and provide inaccurate readings, causing an erroneous OOS event. Third, errors in sample preparation or using incorrect standards can lead to misleading results. These lab errors need to be investigated and corrected to ensure the accuracy of future tests. Category 2, lack of method precision, occurs when a test method inherently has too much variability, causing results to sometimes fall outside the specified range due to random chance. Here are 3 examples. First, a titrometric assay might have a wide margin of error, causing individual results to fluctuate significantly and occasionally appear OOS, even if the product is actually within spec. Second, a spectroscopic analysis could have limitations in its precision, especially for low concentrations, leading to variable readings that might fall outside the acceptance criteria. Third, an assay with complex procedures might have inherent variability, where slight differences in technique between analysts, or even the same analyst performing the test multiple times, can result in OOS outcomes. In these cases, the focus is on improving the test method's precision rather than concluding the product is faulty. Category 3, non-process-related error in manufacturing, involves mistakes made during the production process. Here are 3 examples. First, an operator might forget to add a crucial ingredient to a batch, leading to an OOS result because the formulation is incorrect. Second, a piece of manufacturing equipment, like a mixer or granulator, could malfunction, resulting in a non-uniform product that doesn't meet the required specifications. Third, cross-contamination between different products during manufacturing can lead to an OOS situation, where the final product contains the wrong components or impurities. These operator and mechanical errors typically require investigating the manufacturing process and implementing corrective actions to prevent similar issues in the future. Category 4, process or manufacturing problem, deals with issues arising from poor control over the manufacturing processes. Here are 3 examples. First, incorrect mixing times during a production batch can lead to a non-homogeneous blend, where the active ingredients are not evenly distributed, causing OOS results in the final product. Second, inadequate control over process parameters, such as temperature or pressure, during a specific manufacturing step, can affect the product's quality and lead to OOS conditions. Third, poor control over the blending process, resulting in heterogeneity within a batch, means that different parts of the batch will have different concentrations of the active ingredients, leading to OOS results in some samples. These manufacturing problems indicate potential issues with the overall production process and may require significant adjustments to ensure consistent product quality. Investigating out-of-specification results is critical for pharmaceutical manufacturers, and regulations are quite strict about it. Companies must have clear, written procedures for handling any results that don't meet specifications. The rules stipulate that any single OOS result must be investigated thoroughly and cannot simply be discarded without proper justification, such as clear evidence of a lab error. It's also important to avoid the practice of simply averaging a failed result with passing results to achieve a passing average, as this can be interpreted as manipulating the data. All OOS events must be investigated quickly, and the findings, conclusions, decisions, and corrective actions taken must be thoroughly documented and kept as official laboratory records for that specific lot. This ensures accountability and compliance with regulatory requirements. That's a crucial step in the OOS investigation process. Understanding assignable causes is essential, because if a clear and readily apparent assignable cause is identified, the initial OOS result can be invalidated, and the test can be repeated. The examples you provided are excellent illustrations. If the sample was not representative or insufficient, or if there were errors in the test method or documentation, or if there was an analyst error, or if the equipment malfunctioned, these can clearly explain the OOS result. By identifying these assignable causes, the laboratory can ensure that the investigation focuses on genuine product issues rather than errors in the testing process. This step is particularly relevant to Category 1 and 2 OOS events, where lab errors or method precision issues are the likely culprits. Step 2, conducting the initial investigation, is crucial for determining the root cause of an OOS event. While it can be difficult to pinpoint the exact cause with certainty, a thorough initial investigation is mandatory and goes beyond a simple retest. Simply retesting without investigation raises regulatory concerns, suggests a lack of process control, and might lead to discarding the original result improperly. Therefore, a laboratory must predefine its standard operating procedures and use an OOS investigation form to ensure a thorough and documented investigation. This initial step involves reviewing analytical records, procedures, calculations, equipment, sample preparations, the testing environment, and potential analyst errors. The main goal is to determine if an assignable cause is readily apparent. If thus a cause is confirmed without a doubt, the original OOS result is invalidated, and the test is repeated to obtain a valid result. Step 3, conducting a formal investigation and measurements, is necessary if the initial investigation cannot invalidate the OOS result, or if there are multiple OOS events. Here's how it proceeds. 1. Initiate a full-scale formal investigation involving management, QA, and QC personnel. 2. Extend the investigation beyond the laboratory to include the production area. 3. Review the batch record and any processing deviations to formally identify the source of the deviation. 4. Decide whether to resample if there are not sufficient original samples available. 5. Conduct the formal investigation to identify process or non-process related errors. 6. Have QA management review the investigation, draw conclusions, and propose corrective actions. The formal investigation involving Category 3 or 4 events requires a structured approach, following a specific protocol. The investigation team must first state the reason for the investigation, then summarize the process sequences that might have caused the problem. They need to outline corrective actions to save the batch and prevent recurrence, and also list other potentially affected batches or products, detailing their investigations and any actions taken. This includes examining batches produced by the same employee or machine, or those affected by the same process. Finally, the comments and signatures of all personnel involved in the investigation must be preserved. Step 4A involves repeating the test when an assignable cause has been identified, during the initial or formal investigation. In this case, the original OOS result is invalidated, and all invalidated tests should be repeated, to replace the original results. If the assignable cause was not due to sample preparation, the original sample preparations are used for the retesting. A single reanalysis replaces each invalidated result. For instance, if an assignable cause is found for one of the 10 results from a content uniformity test, only that single result is invalidated, and one additional dosage unit is tested to replace it. Step 4B involves conducting a retest when no assignable cause is identified. A carefully designed retest protocol is needed, based on the specific problem, product history, method, and batch. The protocol must specify the number of retests, typically a minimum of 3 for most samples, and 5 for formulated products. Retesting is done on the same sample set that produced the original OLS result, and a control sample may be used to verify accuracy. Assigning a different analyst is often recommended. Indefinite retesting is not allowed, and the retest protocol must specify the number of retests. After retesting, a decision is made to accept or reject the batch, and further retesting should not be conducted solely to achieve compliance. The FDA guidance on retesting after an OLS investigation emphasizes specific conditions. Retesting is acceptable if the analyst's error is indicated, requiring limited retesting, and potentially replacing the original result. It's also acceptable if the investigation is inconclusive about the OOS category, helping determine if the result is an outlier. Retest results should supplement the initial results in Category 2 when there's uncertainty about method precision. Resampling is permissible only when specified by official monographs, or when credible evidence suggests the original sample was unrepresentative, and this must be supported by solid evidence.
In conclusion, encountering out-of-specification results can happen for various reasons, and addressing them promptly is crucial. An initial investigation must be conducted to identify any assignable causes, such as analyst errors, instrumental issues, test method precision, or calculation errors. If an assignable cause is confirmed, the original OLS result should be invalidated. If no assignable cause is found, a formal investigation must be conducted beyond the lab. Retesting or retesting the same sample helps confirm the original result. It's crucial to maintain all documentation from the investigation for future regulatory audits. The investigation record should include a clear statement of the reason for the investigation, a summary of potential manufacturing causes, documentation review results, history of similar issues, and corrective actions taken.